As a young postdoctoral researcher, Emeritus Professor Peter Koopman took on a project that would define the course of his career. He joined a team at the National Institute for Medical Research in the UK on their search for a gene responsible for developing a fetus into a male.
At that time, we knew it was to do with the Y-chromosome, but we didn't know what it was about the Y-chromosome that makes males.
Professor Koopman's collaborators working in human genetics were able to narrow the search further to a tiny fragment of the Y-chromosome, where they identified a likely gene they termed SRY.
It was certainly a eureka moment, but of course then the question becomes, how do you prove that you have the right gene? And that was where our research in the laboratory that I was working in became significant.
By introducing the mouse SRY gene into genetically female mouse embryos, Professor Koopman's team were able to generate male mice, demonstrating that it was the only gene from the Y chromosome required to trigger male development.
As a developmental biologist, this was a significant result because we finally had the key that could unlock the pathway to studying sex development from a molecular genetic point of view. We now had a starting point from which we could expand the pathway and learn more about how sex develops in the embryo.
Deficits at any point in that pathway can result in sex development variations in children. So it's research that could help individuals and families better understand the genetic causes of these variations and how to manage them. Professor Koopman says research in this field also has applications in other areas, such as ecology, pest management, and agriculture. One example is the work his lab did towards genetically engineering female cane toads to produce only male offspring.
Each female toad is able to lay 20,000 eggs at a time. And so if one can limit the proportion of females in the population using genetic strategies, it may be a way of slowing the spread of pests such as cane toads.
But Professor Koopman's expertise isn't limited to sex development.
The discovery of SRY was a springboard for me to discover other genes that were important in other developmental organ systems.
These genes are part of a broader family of developmental regulatory genes known as SOX genes.
I was able to isolate a handful of other SOX genes, and the study of those genes turned out to be tremendously fruitful because those genes turned out to be key regulators of some other very important organ systems.
Professor Koopman's fundamental discoveries have helped lay the groundwork for further research into the causes, diagnosis, and management of human developmental diseases.
From the outset of my career, I really had it in the back of my mind to try to make a contribution, if possible, to the understanding of human genetic disease. And I'm very fortunate to be able to say I was successful to some extent in that aim.
For his outstanding career in developmental biology, Professor Koopman has now been awarded the Suzanne Cory Medal from the Australian Academy of Science.